It's a very great pleasure to introduce David Page to you today. David is the director of the Whitehead Institute, which he has been since 2005. And David is in many ways an MIT lifer. David, after his MD from Harvard Medical School, became a Whitehead Fellow and then has just stayed on. David is the world leader on understanding the Y chromosome and on understanding sex differesences, not just through hormonal influences, but throughout the body in multiple ways. We was, as part of the Simon Center for the Social Brain, with a focus on understanding autism, are very interested in understanding why is it that autism is 4-to-1 boys to girls in terms of the diagnosis of, formal diagnosis of autism. And it's taken us a few years, but finally, we have connected with David, and what David studies is deeply relevant for understanding autism. So we are delighted that David has become interested in understanding how the brain might become sexually dimorphic in a deep way. And of course, this has evolutionary as well as developmental overtones. And so David's focus on understanding fundamental differences between males and females in health and disease beyond the reproductive tract is of great importance and relevance. David has received many honors, starting, or at least the most prominently starting with the 1986 MacArthur Prize Fellowship. And then many, many other awards and honors and named lectures. He's a member of the National Academy of Sciences. He's a member of the National Academy of Medicine. He's a member of the American Academy of Arts and Sciences. And it's a very great pleasure to welcome David to the Simon Center.
Okay.
Lunch series.
All right. Thank you all, and thank you, Meganker, for the invitation. It's indeed, I'm thinking about a host of disorders of the brain and across the body that show sex biases in their incidence and severity. And I will actually claim that such sex biases are not the exception in medicine. They're in fact the rule. If only we scratch the surface of almost every disorder, we find a sex bias in its aspect of its incidence or its manifestations or its response to therapy. And so what I want to do today is to invite you to join me in envisioning a future medicine where we have a deep understanding of human male biology, human female biology, and where we understand where they are the same, essentially the same, and we understand where they are fundamentally different. And that is some ways off in the future, but what I want to do today is to invite you to join me in thinking about such a future. So, and as Meganker suggested, I think about the future by thinking about the past, that is, by thinking about evolution. And so when I'm going to pose to you the question today, do males and females read their genomes differently? I will suggest that nothing in biology makes sense, Jansky said, except in the light of evolution. And so I'm going to give you sort of a primer on how to think about sex differences from an evolutionary perspective. And I'll start with two images here that I will discuss in some detail, just to get you thinking. So on the far left, I have my favorite pair of chromosomes. To the far left, stately and upright, the rather statuesque X chromosome, and immediately to its side, with its head down, the demure and diminutive chromosome. Now, truth be told, I've spent the better part of my career defending the honor of the Y chromosome in the face of innumerable insults to its character, its future prospects. And I have to say, on behalf of the Y chromosome, what an honor it is, given what has been said about the Y chromosome and those who carry it, to be invited to speak at a center on the social brain. So, and then to the right, I show you a pair of turtles. And I guess that you might not be very good at determining the sex of a turtle in the wild, but this is just to indicate that they nonetheless reproduce without difficulty. And we'll be talking about the turtle in the context of our own evolution. So, where did all of this come from? Where did sexual reproduction come from? So let me set the stage very broadly. So how old is sex? And I'm going to force you, even though you're pining for your lunches, I'm going to force you to actually engage with me verbally. So how old is sex? Well, let's see. Maybe I should come up with easier questions. Like, how old is the universe? Any?
Roughly 14 billion.
14 billion years. Okay, fantastic. Thank you. So how old is our planet?
4.5.
4.5 billion. So if our planet is 4.5 billion years old, then I'm going to guess that sexual reproduction had its beginnings. The first great invention in the move towards sexual reproduction as we know it today was sexual recombination, and I'm going to credit prokaryotes with having devised a scheme of exchanging hereditary information on the order of 3 to 4 billion years ago, to the age of our planet. But it turns out that in prokaryotes, the exchange of hereditary information is a fairly free-form, unrestricted, uninhibited activity. All of this changed with the coming of the nucleus and the appearance of eukaryotes, I'm sure you all had some introductory biology course where you were first introduced to the difference between prokaryotes and eukaryotes. And I'm going to tell you something that you were not told, but you absolutely should have been told, that a defining feature of eukaryotes is that they put their genes in pairs. Now, why are genes in pairs in eukaryotes? Of course, so we can teach Mendelian genetics. There would be massive unemployment of geneticists on this campus and across the world if genes did not come in pairs. No, genes come in pairs, so we pairs apart at meiosis and put them back together again at fertilization. And so that means that the life cycle has an alternation of haploid and diploid phases. This is all coming back, all this biology is coming back to you, right? And I put up here as a poster species for taking sex to this level. Anybody recognize this organism? Yeast. Okay, fantastic. And this little thing here, which has got 4 parts to it, is a yeast tetrad. It's the 4 products of a yeast meiosis. Here we're just staining for DAPI, for the DNA content. And it turns out that meiosis appears to have been invented once in the basal eukaryotes, on the order of 1.5 to 2 billion years ago. So in fact, this most central of sex acts, that is meiosis, is actually about 1.5 to 2 billion years old. And we and yeast share fundamentally the same machinery of dividing our chromosomes, or dividing the number of chromosomes, dividing the number of genes in half. So, but the trick is with yeast, there are 2 kinds of spores in yeast. Anybody know what they're called? They're imaginatively referred to as A and alpha because you can't tell them apart structurally. So A and alpha spores are structurally indistinguishable. So the next invention that gets piled on top of genes in pairs, that gets piled on top of meiosis, the next invention is gametes. So making one of the spores big, making one small, and now we start to call them egg and sperm. And actually, whoever makes the big gamete, we will refer to as a female, and whoever makes the little gamete we'll refer to as a male. This is, in fact, the most generally applicable definition of male and female across the animal kingdom. Do you make the big one or the little one? And it turns out that this was invented, it evolved several times. In our lineage, this appeared approximately 600 million years ago. So again, I'm at the point that dimorphic gametes are evolved on top of the more fundamental, more ancient innovation of meiosis itself. So, and I will offer up as a poster species for taking sex to this level, I will offer up the turtle. Now, why the turtle? Does any sex is determined in how the sex of a turtle is determined?
Temperature of incubation.
Temperature of incubation. So you're doing really well. I will have to buy you lunch. Right. You know, you're doing very well. So it turns out that, you know, turtles lay eggs. And it's actually the temperature at which the egg incubates that determines whether the turtle will develop anatomically as a male or a female. So what this means, temperature-dependent sex determination means that there are no sex chromosomes. In fact, male and female are genetically identical. I want you to let this sink in a little bit, because what it means is that in turtles, the existence of two sexes is purely epigenetic. Now, I think we're more conventionally familiar with thinking about epigenetics as something that tweaks something a little bit. Well, here I'm saying that epigenetics leads to having a male anatomy or a female anatomy. And I offer up here some line drawings of simply the reproductive tracts of male and female turtle, and I will claim that those male and female reproductive tracts of a turtle are every bit as distinct as those of a mammal. But there are no sex chromosomes. This is purely epigenetic. So what that means is in turtles know how to get have two different readings of the same genome. So when I ask the question at the beginning, do males and females read their genomes differently? If I were talking about turtles, I would say absolutely. They absolutely know how to read their genomes differently. And I will suggest that we have not forgotten what turtles still know. We too know how to read our genome ways, but we have gone on to a fourth invention that gets piled on top. So if a turtle is a representative species for taking sexual reproduction to this stage, let's now add the fourth invention, which is sex chromosomes. Which, if it's the male that has two different sex chromosomes, they're called X and Y. And just so you should know this at every cocktail party to which you go, if it is the female that has two different sex chromosomes, then they are referred to as Z and W. It's just a naming convention. And species that have ZW sex chromosomes include butterflies and birds, for example. XY, of course, is mammals and flies and so on. And it turns out that sex chromosomes, as I will argue, sex chromosomes have evolved independently many, many times across the animal kingdom and actually across the plant kingdom. Our own sex chromosomes are the oldest documented sex chromosomes at about 300 million years of age. So the point I'm making is that overall is that in this evolutionary scheme, the sex chromosomes are a late addition to an already perfectly functioning system of meiosis, dimorphic gametes, and male and female. And I hope you notice that the order in which I'm presenting these things is exactly the opposite of the way sex determination and sex differentiation is taught in every university and every medical school in the world, where the lecture always begins with what? The sex chromosomes, right? And then everything flows down from them. Well, that is true in some sense, in a developmental sense. But in an evolutionary sense, the sex chromosomes are absolutely a Johnny-come-lately to the system of two sexes. Okay, so where then did our sex chromosomes come from? Well, it turns out we parted company with the ancestors of birds about 325 million years ago. So this poses an interesting quandary because in mammals we have an XY system. In birds, they have a ZW system, but we have a common ancestor 325 million years ago. So how could this be? And so I'll just say the male-specific Y is in some sense the reciprocal of, in birds, a female-specific W chromosome. It's a sex-specific chromosome, a sex-shared chromosome. X, mammals, Z, birds. So how did this all come to be? Well, over the last 20 years, actually, mostly through the work of a series of grad students in my lab, we've had a lot of fun reconstructing and becoming convinced that your X and Y chromosomes evolved from a perfectly ordinary pair of autosomes that existed in our common ancestor with birds. And one of the strongest pieces of evidence to support the claim that our X and Y evolved from ordinary autosomes is that those chromosomes still live on autosomes today in birds. Okay, so next time you go to a chicken farm and you are taking samples for sequencing, and you'll have some, sequencing device about this size, you sequence the chicken, and you then will get out your smartphone and do a sequence alignment with your own genome. You will find that your X chromosome is a dead ringer for chicken chromosomes 1 and 4. Actually, the short arm of your X chromosome is chicken chromosome 1. The long arm of your X chromosome is chicken chromosome 4. Now, I'm only slightly oversimplifying. These are really good matches. No mistaking it. So where then did the bird Z and W chromosome come from? Well, it turned out the Z and W in the chicken and all other birds evolved from different ordinary autosomes of our shared ancestor. And guess what? Those autosomes that gave rise to chicken Z and W live on in us as autosomes. So we have these two beautifully reciprocal experiments of nature. I mean, every time I show this and think about it, I sort of stop. I'm stopped by the, the gorgeousness of this from a common starting point. And then I will just add, most recently, we've actually been looking at the snake sex chromosomes, which are also ZW. And we also share with them a common ancestor. And they represent yet another independent foray into this set of ancestral autosomes, giving rise to an independent ZW system in snakes. So now we thought that, so given this sort of symmetry, the question was, could we understand the nature of the genetic differences between male and female in our own species better if we could reconstruct with great precision these ancestral autosomes that gave rise to XY or that gave rise to ZW. So what I'm going to now tell you is that we've been able to reconstruct in silico. Now, I should say, these ancestral autosomes, they do not exist today. They existed 300 million years in the past. But it's possible through comparative genomics to electronically reconstruct a reasonable image of those sex chromosomes, or at least to reconstruct the gene content of those ancestral autosomes. And so Winston Blott and others in our lab set out some years ago to do just that. And here I'll just show you the pictures of people who have contributed to this effort, especially over the last decade. Up top, Winston Blott, Jen Hughes, and Helen Skoletsky, other colleagues at Whitehead, joined in a massive sequencing effort by Rick Wilson and colleagues then at Washington University, and Richard Gibbs and colleagues at Baylor. And I'll tell you what came of that. So construction of the ancestral autosome has gone pretty far. So first I'll tell you about, I'm going to show you in parallel here 2 experiments of nature. Reconstructing the autosomal ancestor of X and Y, reconstructing the autosomal ancestor of avian Z and W. And it turns out that Winston and colleagues have shown that ancestral autosome that gave rise to your X and Y chromosomes carried about 639 genes. The chromosomes that gave rise to the chicken sex chromosomes carried about 685 genes. And I put these up as a true Venn diagram. So these are non-overlapping sets of genes. The completely independent origins of the mammalian sex chromosomes and of the bird sex chromosomes. But you see, starting with comparable numbers of genes. So let's see what happened then. Well, it turns out that over on the mammalian side, so I should say, again, fully independent experiments of nature. Over on the mammalian side, the human X chromosome, of those 639 ancestral genes, today's human X chromosome retains of those 639 about 629. So in other words, the human X chromosome today is pretty much a dead-on ringer for the ancestral autosome that gave rise to the X and the Y. Now, at least 629 of those 639 ancestral genes survive. Now, what I'm not going to tell you about today is that the human X chromosome has also acquired a bunch of genes that were not on that ancestral autosome. Now I'm going to focus on genes that were retained from an autosomal ancestry. So that's what happened on the X. Let's see what happened over here on the Z. Well, it turns out the chicken Z, today's chicken Z chromosome of 685 genes, retains 667. So very similar sort of outcome in a sense. Now let's see what happened to the sex-specific chromosomes. Over on the mammalian side, the Y chromosome, much smaller, retains only 17 of those ancestral genes. Now, those 17 genes that survive on the human Y, all 17 also survive on the human X. So what I'm going to be telling you about are then 17 XY gene pairs that persist from the autosomal past. And I'm going to suggest that those 17 persisting genes may have a great role in our thinking about sex differences across the body. But let's see what happened over here on birds. So it turns out that the female-specific W chromosome did a slightly better job than the male-specific Y of maintaining ancestral genes. The W in chickens retains 28 ancestral genes. And all 28 also survive in the chicken Z. So again, there are 28 ZW pairs in birds that are in some sense the mirror image of those 17 XY pairs in mammals. Well, I'm going to now more or less end the conversation regarding comparative studies of birds and mammals and just drill down now on the human X and Y. So how did this all come to be? I mean, thinking about the X and the Y, if the ancestral autosome had 639 genes and the human X maintains 629, how is it that the Y ends up with just 17? How does this work? Well, here is our understanding of how it all unfolded. So again, 300 million years ago, when we were reptiles, we probably had temperature-dependent sex determination. You know, and if you get together with family at Thanksgiving or other holidays, you can talk about the old days, the old times, and if your family records go back that far, you will remember the days of temperature-dependent sex determination. But something happened then. A mutation arose to begin as to what would live on today as the sex-determining SRY gene in the Y chromosome. And then what unfolded was that first in the immediate vicinity of SRY and then over an expanding region, the Y chromosome began to develop isolationist policies. It decided to stop exchanging goods with the X chromosome. Now in genetics we call this recombination, but it's, you know, the economic analogy is perfect. So it's basically a shutdown of it between the X and the Y chromosomes. Now, the X chromosome continues to enjoy the benefits of free trade in what setting? In female meiosis, right? So in the making of eggs, you still have two X chromosomes recombining. So what we're talking about is a shutdown of trade between the X and the Y chromosomes, first immediately in the vicinity of SRY, then over a spreading region in male meiosis. And this, of course, led to an immediate decline in the economy of the Y chromosome and the loss of many, many genes from the Y chromosome. And so it goes on to include the bulk of the Y chromosome is eventually in this no trade zone with the X. Loss of many, many genes. Meanwhile, the X chromosome is largely retaining the gene content of the ancestral autosome. Okay, so if you extrapolate this out to the right, it's not a pretty picture for the Y chromosome, right? And we were not the only ones to think about this. So here was an editorial that was published 15 years ago in grandly titled The Future of Sex. And in this, my colleagues John and Jenny relayed what I just told you, that the original Y chromosome contained, well, I said it was what? We said it was 639 genes, 1,500. It's pretty close. But during the ensuing 300 million years, all but around 17 were inactivated or lost. And then they did something quite spectacular statistically, which was to take 2 points and drew a straight line through 2 points, because what else could you do with 2 data points than to draw a straight line through them? And so they went on to predict, based on this, that at the present rate of decay, the Y chromosome will self-destruct in around 10 million years. I'm not making this up. They published this in Nature. And they did not actually explain their methodology, but we have done a re-engineering of this and deduced that they drew a straight line through 2 data points. Okay. So the question then is the Y chromosome actually presently decaying? Well, so to answer this question, we thought we needed some more data. So we appealed to some close collaborators here and decided to compare the Y chromosome of, in this case, Stephen, with the Y chromosome of a chimp and a rhesus monkey. And you know that we and chimps are separated by about 7 million years, and we and rhesus monkey are separated by about 25 million years. And actually what turned out from really full sequencing of these Y chromosomes was that the human and the rhesus Y chromosomes carry nearly identical sets of ancestral genes. So I said that humans retain 17 genes. Turns out rhesus retains 18 genes, and it's almost a precisely overlapping set with those retained on the human Y. The implication is that in the bulk of the human Y, gene loss had ceased essentially prior to our divergence from the rhesus monkey. And the same goes for the rhesus lineage. So, and by adding additional species, we came to the conclusion that what had actually happened during the evolution of the chromosome was that initially there'd been a very rapid exponential loss of genes from the Y. A calamitous loss in the first few million years, but that had given way to strict conservation through purifying selection of a much boiled down Y chromosome. So, where did that, so how to think about this? How to think about these surviving genes? So I'll just give you an example here. And now we're beginning to turn our minds towards what are the implications here for human biology. I'm just going to show you an example here of one of these surviving XY pairs. It's KDM5C on the X, KDM5D on the Y. These XY genes are not, I want to make the point that they are similar but not identical. Some of you may have heard of genes on the X and the Y that are called pseudoautosomal. There's a pseudoautosomal region actually up at the very tips of the X and the Y, where XY recombination is a normal and frequent event in male meiosis, and there the X and the Y are absolutely identical. I'm talking about regions of the X and the Y that do not ordinarily recombine, and so they have had time over the tens or hundreds of millions of years to diverge. So here the DNA sequences of X and Y, even within the coding region of this gene, are only 85%. They encode proteins in this case that are 87% identical at the amino acid level, so you could call them isoforms. And the X and Y genes are both expressed in human tissues. That means that XX tissues express exclusively the X isoform. And I will say, for those of you not familiar with it, this is a very sexy protein. This is an H3K4 demethylase, a central player in gene regulation across the genome. And then XY tissues express both the X and the Y isoforms. And this is, I'm giving you just this example, but there are 16 other XY pairs where I could show you the numbers. And for most of these XY pairs, they have no known role in sex differentiation as classically defined. So what do I mean by classically defined? The classic perspective on sex differentiation in mammals focuses on the reproductive tract, the anatomy of the reproductive tract. Sort of what you had in 7th grade health class is sort of what is, that's the classic definition of sex differentiation. Now, when I say this is true, no known roles in sex differentiation for most of these XY pairs, there's one glaring exception, and that is SRY. And it has an X chromosome counterpart called SOX3. Well, I'm making the point here that even SRY, the sex-determining gene on the Y chromosome, derives an ancestral autosomal gene that was present on that ancestral autosome that gave rise to X and Y. But what I'm going to suggest is that maybe we need to think about, maybe we need to expand our understanding of sex differentiation and sort of all of health and disease into an expanded understanding. This is a survivor. So what I'm really telling you is that the voyage of the Y chromosome is not unlike the voyage captured by this artistic rendering. And of course, I can't help when I look at this image and I see the Titanic on this steep downward trajectory, I can't help but be reminded of that line that was drawn through 2 data points. And of course, your eye is drawn first to the sinking ship, but my eye is actually drawn to the foreground where the life rafts are, and there are the survivors, all 17 of them. So let's say a little bit more. Let's say a little bit more about those survivors. And just as in the case in the storytelling around the voyage of the Titanic, questions arise like, were the genes that survived in the Y lucky? Were they in first class? What was, why did they survive? Did they have any special qualities that favored their survival? And I will tell you that we did not seriously entertain this question until the last 3 or 4 years. And seriously pondering this question has completely changed my way of thinking about sex differences across the body. So, what are the implications for male and female biology? So I will say that our view of this is now informed by complete sequences of the Y chromosomes of these 8 mammals. I've already told you about our own Y together with the chimp, and we've added the marmoset, the mouse, the rat, the bull, and for good measure, we threw in one marsupial. This is the South American opossum, which is a laboratory species. And as an outgroup, I add the chicken, whose Z and W chromosomes we've already mentioned. And what came out of this, so we have essentially complete sequences of the ancestral regions, very accurate, very contiguous. First systematic analysis of ancestral Y genes beyond primates. And in aggregate, if we add up all of this evolutionary branch length, we're looking at a billion years of mammalian sex chromosome evolution. And I'm going to suggest this casts our understanding of the Y biology and X biology in a new light. So here, I'll just give you a sort of traveler's guide to the Y chromosomes of these 8 mammalian species. So the species are all listed up top here. Here's the opossum through the human, phylogenetic tree that connects them. And down along this list, here are listed 36 genes that survive today from the ancestral autosomes, 36 genes that were present on the ancestral autosomes and that survive on the Y chromosome in one or more of these 8 species. So out of the 639 genes that set sail on the Titanic, there are only 36 that survive today on the Y chromosome of one or more of these 8 species. And where a black dot is present, it simply means that gene is retained on the Y chromosome in that species. So it turns out all 36 genes also survive on the X chromosome. So if you were to aggregate all 8 species, we're talking about 36 XY gene pairs. And comparisons across species indicate that gene survival was non-random. Something that's quite remarkable is that actually 2 of these genes, UBE1Y and KDM5D, I've already mentioned, it turns out that 2 of these genes actually survived this ordeal twice, independently. It turns out that the evolution of the placental mammalian and the marsupial Y chromosomes they started out together, but the separation of these 2 lineages 130 million years ago means that there's been these are substantially independent ongoing experiments of nature. It turns out that these 2 genes survived the ordeal twice, which is to say, it is if they got on the Titanic, they made it across, they got on the life rafts, they survived, they went and did it again, and they survived a second time. This does not happen by chance. But we came to understand other things about these surviving genes. So it turns out the 36 ancestral XY pair genes have special qualities versus controls. Now I got to explain to you that nature provided us with the controls for this analysis. Since we have reconstructed the passenger list on the Titanic, we know exactly who went down with the ship, okay? So all those genes that failed to survive now serve as the control group for considering the qualities of the survivors. And what we see is that the survivors, compared to those who went down with the ship, the survivors tend to be expressed in adult tissues. They are more widely expressed across the body, less tissue specific. They tend to also be expressed in pre-implantation embryos. So they're joining a sort of select company of genes broadly expressed in the adult, also expressed in the embryo. And by a variety of measures, they show a high likelihood of haploinsufficiency. Now, haploinsufficiency is the scenario where you have a phenotype, you're in trouble if you have just 1 copy of the gene as opposed to 2. So being haplo is insufficient. And interestingly, the X homologs of these survivors on the Y escape X inactivation. So you all know about X inactivation. It's the silencing of the second X chromosome in a female's cells. But it turns out that there's a substantial number of genes on the second X chromosome that are transcriptionally active. And that is essentially the case for the X homologs of all of these survivors. Okay, so this is pointing to dosage sensitivity of these genes. And in fact, we think that these XY pair genes probably play a really critical role in the Turner syndrome phenotype. Now you'll remember, you all got taught about Turner syndrome in your first biology class. It's XO, it's girls and women who have a single X chromosome and have a variety of, are infertile, webbing of the neck, short stature, a whole variety of signs and symptoms. And we think that actually Turner syndrome is the result of having 1 copy rather than 2 copies of these haploinsufficient genes. And it turns out that these genes also look to play, are highly enriched, the surviving genes are highly enriched for genes that have central roles in gene regulation and expression. And let me show you what I mean by that. This is just a sort of blob chart of the categories of function of the surviving XY gene pairs. And now here I'm taking not just the 17 that survive on the human Y and X chromosomes, but I'm taking this is encompassing most of the 36 XY pairs that survive in 1 or more of those 8 species. So we can have a larger set to see more richly. What we see is that many of these XY pairs play critical roles in chromatin modification. I told you already about an H3K4 demethylase. There's also an H3K27 demethylase. Chromatin modification, transcription, splicing, translation, protein stability, and degradation. So central aspects of gene expression are highly enriched among the surviving genes on the X and the Y chromosomes. So this leads us to what I would say is the new understanding of the human X and Y. So the old understanding, prior to this work, the view was that the Y chromosome functions only in the testis. We now understand that, actually, and I didn't go into all the details, of the 17 survivors on the human Y chromosome, 12 of them are very broadly expressed across the body. So 12 different Y genes are expressed throughout the body, and they encode central players in gene regulation and expression, about as central as you could ask for. And then the old idea was that in females, the second X chromosome is transcriptionally silent. But what we now know, and I haven't gone into this in such detail, is that actually there are hundreds of genes on the X chromosome that are transcribed from the second so-called inactive X chromosome, and these include X-specific versions of the Y's broadly expressed genes. So if you take these two old ideas and you put them together, you would arrive at the notion that outside the gonads, that X, Y, and XX cells are functionally equivalent. I want to stop and let you ponder that for a moment, because this is in fact how most of the world thinks about this. And why would that be the case? Well, if the second X chromosome is transcriptionally silent, then female cells are functionally XO. If the Y chromosome functions only in the testis, then male cells outside the testis are functionally XO. This is the way the world continues to think about this. But what I'm saying is, if we now add this new understanding, we begin to realize that throughout the body, XY is not equal to XX. And let me take all of what I've said and turn it into one cartoonish slide, which is that I would say this is the way sex differentiation is taught today in all universities and medical schools around the world. That whether you're XX or XY is of direct and immediate consequence only in the nether regions, only in the gonads. And then the hormonal secretions of the testes or the ovaries masculinize or feminize the rest of the anatomy, the rest of the body. But what I'm suggesting instead is that there, as a direct consequence of the evolution of the X and the Y from ordinary autosomes, that there are in fact subtle but consistent biochemical differences between XX and XY cells across the body before they ever are exposed to sex hormones. Now, I don't mean to actually replace this idea with this one. I mean to hybridize them, like so. So in other words, whether you're XX or XY is of indisputable significance in determining whether the fetal gonad becomes a testis or an ovary. And that in turn has an enormous impact on the subsequent sex hormonal environment. But there are sort of pre-existing biochemical distinctions between XX and XY cells. So this leads me to where we're going and where my lab is going now, and where I'd like to invite you to join us if you'd like. So we're pursuing this hypothesis that in most tissues and organs, XX and XY cells differ biochemically as a consequence of their evolution from ordinary autosomes. And the long-term goal would be to understand the roles of sex chromosome constitution, and specifically perhaps of these XY gene pairs, but I really mean it very broadly, to understand the roles of sex chromosome constitution and sex differences in health and disease across the body. And I will suggest we need to understand this not in a fully human-centric way, but in a broadly comparative, multi-species perspective. So why does this matter? Well, in a room of people who are thinking about things like autism, I don't think I have to say, but let me broaden let me make the case nonetheless. So with a disease like rheumatoid arthritis, for every man who is affected with rheumatoid arthritis, there are about 2 or 3 women who suffer from this disorder. As Magankar said at the beginning, with respect to autism, for every girl with an autism spectrum disorder, about 4 boys. I'll flip it back again. Lupus. For every man with this devastating disease, there are about 6 of them. And even when, and I could go on and on and on, and even when the incidence of the disease is similar in male and female, the manifestations can be quite different. I just give an example here from my good friends Johnny Cricket Seidman at the Brigham and Women's Hospital looking at a disorder called dilated cardiomyopathy. This is where the heart wall thins and the heart balloons in a very dangerous way, typically leading to an early death, a death in adulthood. And I'm just going to show you here the survival or the death curves, if you will, for women, men who have essentially the same genotype. So this is the survival plot for women. And men with morally equivalent genotypes tend to die about 10 years earlier. Actually, when I saw John Seidman present this at a human genetics conference in San Diego about 5 years ago, this really caught my attention. And I asked John, after he projected this, I said, John, well, since you showed this, you must have some ideas as to what accounts for this sex difference. And he said, I have no idea. I showed this slide so you would ask. And he hooked me with that. He has no understanding as to why this sex difference occurs. So, but where could it be? Well, and what's the issue? I'm going to say that the human molecular genetics that we have constructed, actually much of it, right here on this block, the human molecular genetics that we have constructed, together with scientists from around the world, is a unisex conception. And it was captured, I'll go back to Bill Clinton's State of the Union address in the year 2000. Towards the very close of that State of the Union address, said this fall at the White House, Hillary, you've heard of her, had one of her Millennium dinners, and we had this distinguished scientist there, I will not name any names, who is an expert in this whole work in the human genome, and he said that we are all, regardless of race, genetically 99.9% the same. Now, you can understand why this was, for Bill Clinton, a politically very attractive framing, right? Same. And the trick is, the statement is correct as long as the two individuals you're comparing are both males. It is also correct if the two individuals you compare are both females. But as we have been discussing, the problem is if you are so unfortunate or misguided as to compare a male and a female, then the difference balloons by a factor of 15. Or if I just take if these are the percentage identities, I now flip them around in terms of differences. If we go from a 0.1% genetic difference between randomly selected males on the planet or between two randomly selected females on the planet, we have a 1.5% difference between a randomly selected male and a randomly selected female. Why does this matter? Well, up top, we have now an enormous commitment nationally and internationally to something called precision medicine, which will focus like a laser on the significance of those 0.1% differences between two males or between two females. But there's this other thing down here that's 15 times larger called sex differences, which gets barely a nod. That's why I'm here to talk with you today. And when I say, and of course, what is this difference? The difference is to be found in whether you've got two X's, you've got an X and a Y. And as I've explained, whereas we used to think that all of us were sort of genetically, effectively XO outside the gonads, we now know better. And I want to say, we have, throughout the history of human biology, the existence of two sexes has been a tremendous inconvenience. And actually, truth be told, we know that there's some element of that in our own animal studies, right? But let's come, when I was back to the human molecular genetics revolution. So you've all heard of GWAS studies, genome-wide association studies. What you may not know is how the sex chromosomes have fared in this last decade of GWAS studies. So, but I'll first say, how many of you have heard of this thing called OMIM, Online Mendelian Inheritance of Man? It's McCusick's, the thing that Victor McCusick started of phenotypes, and I just make the point here that for the X chromosome, molecular basis known phenotypes, molecular basis unknown phenotypes, 7–8% of all the phenotypes that have ever been cataloged in our species map to the X chromosome. But in the world of G, we go from 8% to a very different picture. I show you here a summary published about 3 years ago of all the statistically significant GWAS hits for all traits, actually for any one of 740 traits that had by that point been cataloged. And each little circle here, each bauble here is a statistically significant GWAS hit mapped to the chromosomes. Now it turns out, here's the X chromosome. In the old days, it used to get confused with chromosomes 7, because they're about the same size. But you see how many baubles and ornaments there are on 6 and 7? And you see the X by comparison? Well, actually, and it's even worse for the Y, of course, nothing over here. So it turns out the X chromosome carries only 0.7%. In OMIM, 8%, but in GWAS, it's 0.7% of all statistically significant published GWAS hits. Now, you could say, perhaps this is because the X chromosome actually makes no biological contribution. It turns out that the X chromosome has been systematically excluded from 90% of all GWAS studies in the past decade, for a whole variety of technical reasons, like most of the GWAS analytics software requires that everyone tested has 2 alleles. So males have only 1 allele, so we'll not do the X. Now, I like to point out that we require that all 7th graders taking their first biology course come to grips with the X-linked recessive mode of inheritance, which also requires that you think about males having 1 allele. But so we have a lot of work to do here. So I'm saying the onrush of this field has, as I sometimes like to say, it's been projected that it might take 10 million years to make the Y chromosome go away. It's taken only 10 years of GWAS to make the X chromosome disappear. So something needs to be done here. And with that, I will, I should say, so I've sometimes, I've suggested that perhaps instead of calling these genome-wide association studies, we should call them neuterome-wide association studies. So I'll close saying that until we arrive at an appreciation of how male and female genomes differ, that is that 1.5% difference, and of how males and females read their genomes differently, to the extent that we remember what turtles still know, we will be surprised time we encounter a sex difference in disease incidence severity or response to therapy. And with that, I would like to issue an open invitation to everyone here and beyond to join us in studying sex differences in health and disease. And I will just quickly list off, to give you a sampler of where my lab and collaborators are going to try to open up this space. Alex Godfrey has been discovering how widely expressed genes on the Y chromosome have facilitated across evolution, have facilitated female specialization of X chromosome expression. Sahin Naqvi has started a really ambitious project to look at sex differences in gene expression across the body, looking in detail at 12 tissues, 5 species. This is with support from Biogen. He's also been looking, again in collaboration with Richard Ranzelhoff at Biogen and Chris Glass at UCSD, he's been looking at sex differences, and this is the project that perhaps comes closest to the interests of this audience. He's been looking at sex differences in the mouse and human brain with a focus on microglia. Lucas Schmattel is looking at sex differences in the human heart. This is in collaboration with John and Cricket Seidman, whose work I've already mentioned, Rick Mitchell at the Brigham and Women's Hospital, and Steve Geige. And Lukáš is already finding very interesting differences in the translational capacity of cardiomyocytes, male and female. Laura Blanton is looking at sex differences in human immune cells. I told you about the autoimmune disorders showing dramatic differences, male and female. This is work in collaboration with Listeners Group at NIH and Andrew Lane at the Dana-Farber. And then in what I might consider a new platform that we're trying to construct, Adriana San Roman, together with Max Munke at NIH and Carol Simango-Sprouse at the Focus Foundation, is taking advantage of of an experiment of nature to alter the number of sex chromosomes. So sex chromosome aneuploidy, like Turner syndrome XO or Klinefelter's XXY, is actually quite common. And in fact, there are people walking around with 3, 4, or even sex chromosomes. These are the only chromosomes in the body these are the only chromosomes of ours that are consistent with viability when taken to such numbers, taken up or down to such a degree. And Adriana is beginning to look and seeing very interesting effects of these changes in sex chromosome constitution on global gene expression. And so with that, I will just throw up a slide of those who've been supporting this nascent work and other work in the laboratory. And I'd be delighted to take questions if time permits. Thank you very much. Yeah. Yes.
So David, my intuition is that if your model is correct, there should have in fact been many instances of hypomorphic mutations in those 12 very essential genes that you've indicated. And this sort of duplication phenomenon also goes against this model. But if you can just sort of have people walking around with, 3 or 4 extra copies of it, doesn't seem to quite fit. So are there diseases that have been linked to these 12 genes that are out there?
So great question. So how can I have it both ways, right? I want to say these genes are really essential. So first, let me point out, so if you go back, so you're sort of saying, if all this is viable, then how can any of this really hold up? Okay, so first of all, let's go to XO, Turner syndrome. So now, having told you about Turner syndrome, I'm going to completely flip it around and say, actually, XO is inviable in humans. It turns out that 99% of human XO fetuses abort spontaneously. And the only ones that survive are probably mosaic for a second cell line that retains a second sex chromosome. Okay, so it's actually an X karyotype is one of the very most common chromosome anomalies discovered in human miscarriages. So, and in fact, we strongly suspect that all the XY individuals in this room are viable only because of the expression from the Y chromosome of these 12 genes. So what I'm saying is the XO condition is essentially a lethal one. So you're quite right. We've got to look through in detail. There are phenotypes.
So from
in
these
these
12 genes,
yeah.
Are there any diseases that have ever been found that are hypomorphic because of some mutation in those 12 genes?
So it turns out that those 12 genes are incredibly, show a much reduced level of variation compared to other genes on the X chromosome in their amino acid sequences, for instance. So what I'm saying is there's actually evidence of incredibly intense selection operating on all 12 of those, if you see where I'm going.
You're walking around with a null mutation on one of those 12 genes.
Well, there would be people who have partial deletions on the X chromosome or partial deletions on the Y chromosome, and those are individuals who we're now going back to look at in more detail. So I see exactly where you're going, which is to say, how can these things be so critical And how can they be so dosage sensitive, and yet you have these? So we've got to explore that more fully, but I'm saying the evidence of incredibly intense selective pressure on these XY there. There have been now quite a few mutations described, somatically arising mutations in various cancers that affect especially the genes on the X chromosome.
I would argue that the alternative model, if my contention is right, that these are not important, is that perhaps the X chromosome version of those 12 genes, because they are not inactivated, are somehow protective in their properties that are lacking in a male. And it's really protection coming from the female versions of those 12 genes that's what's important.
Well, certainly the X copies of these genes are going to be intensely selected, right? Because they're present in a single copy in male, and females, of course, are doing just fine with exclusively the X version. Part of what we're getting at is what are indeed the functional relationships between the X and the Y isoforms for these pairs. And what I want to point out is that it's not just a question of the encoded proteins, the interchangeability of the encoded proteins. And actually, we and others have in a few cases demonstrated the functional interchangeability in cell cultures and such of the X and the Y encoded proteins. For many of them, we don't yet know that. But beyond the question of whether the proteins are functionally interchangeable is the realization that evolution has had tens or hundreds of millions of years to also play with the promoters and enhancers of these genes on the X and the Y chromosomes. And we're already seeing very substantial divergences in regulatory elements on the X and the Y gene pairs, which may shift the level of the relative expression of the X and the Y players on a tissue-specific basis. We're actually already seeing pretty strong evidence of that. So there's a lot of subtlety here. I will completely grant you that. Yeah.
I have a follow-up.
Yes.
Kind of a question.
Yes.
Kind of follow-up question. So how should we think about this in the context of autism spectrum disorders?
Yeah.
How might the genes on the Y chromosome confer risk or protection?
Yes.
On the emerging tally of risk genes for autism?
So great question, and happily I have a definitive answer. No, I'm teasing, right? So the X and the Y, these XY gene pairs, again, are global regulators. There is one of them, one example, in EIF1A, an essential translation factor is encoded by the X and the Y chromosome. There's no protein in the body that is made except the agency of EIF1A. Here, actually, the protein isoforms differ by only a single amino acid. But we find that the regulatory elements on EIF1AY, EIF1AX are highly diverged. So imagine that you had in XX and XY cells of the brain, and I will allow you to pick whatever cell type in the brain you think is most critically implicated in an autism spectrum disorder. I raise the possibility that protein synthesis is tilted a little differently in XX and XY cells as a result of those differences in expression of EIF1A. And I don't have to stop there. I can go to the H3K4 demethylase, H3K27 demethylase that are encoded as XY pairs, and that will be broadly influencing gene expression across the body. So if you think that autism might be influenced by modest changes in the relative expression of different genes or proteins, not all or nones, but modest changes, then I think that the X and Y chromosomes absolutely set the table for entertaining those hypotheses. And I think I think this is also very explorable. I think we simply have to start by cataloging in a multi-omic way the molecular differences between XX and XY cells. And I will suggest that if sort of a corollary of what I'm suggesting is that in those cases, whenever we see a sex bias in the incidence or severity of disease, I will suggest that that implies a sex bias in health. In other words, I don't think these sex biases in disease arise out of nothing. There will be pre-existing sexual dimorphisms at molecular levels between XX and XY cells. And I think we now have ways of contemplating how these things could arise. Now, I also will say the sex hormones are playing their roles. I don't mean to diminish or exclude the notion that androgens and estrogens are, for example, influencing gene activity and gene expression across the body. They absolutely are. But I'm suggesting that they're doing that in concert with and in common with pre-existing molecular differences between XX and XY cells.
Okay, let's take a couple of very quick questions.
Yeah.
And then we'll let David go.
Yeah.
Just about the sort of relative magnitude of the hormonal effect versus the genetic differences that you're describing. And,
Yeah.
Can you give us some sense of what percentage of the transcriptional differences?
Oh, wow.
And also,
Fantastic, yeah.
I would imagine that prior to the differentiation of the gonads, there is no hormonal effect. Have people looked for very early transcriptional differences that could be attributed to?
Okay, so fantastic questions. So your first question was, so if you look in a given adult tissue and you find sex differences between male and female, and I can assure you, you can find sex differences in any tissue, male and female. How many of those, to what degree are those sex differences due to sex hormones, and to what degree are they due to these sort of hardwired sex chromosome effects that I've named? Well, we don't know. We're just beginning to ask that question. I would say the assumption until this point has been that they're all due to sex hormones. So we don't know whether the sex chromosomal effect will be 1% or 10% or 50%. But what we can see already is that we can actually we can sex any given tissue now from its transcriptome. Okay, it's fun. It's turned out to be quite straightforward, and not just because we can find Y-derived scripts. Okay, so we could actually, I think, in several cases, we can determine the sex. You could figure out the sex of a tissue from its transcriptome simply by looking at a modest number of, we can take the autosomes and do it. We take the X chromosome, we can do it. Obviously the Y will do just nicely. So it's an open question. And then you asked, well, what about prior to the sexual differentiation of the gonad in early embryos? We're just beginning to look at that question now. It's a really interesting question because we could look, and then of course the question will emerge also, shortly, at all the stages in sexual differentiation, in the neonatal period, before and after puberty, before and after menopause, all of these will arise. And these questions will also, we think, be worth examining actually in the context now of trans therapy, right? So there are substantial numbers of individuals who are undergoing sort of sex hormonal therapies to align more their bodies with their minds. And that also represents an interesting opportunity that we might explore. Yeah.
Okay, Dara, last question.
Well, this is back to the previous conversation.
Yeah.
So in disorders that have an imbalance between the sexes, such as autism,
Yeah.
does that mean, so the Y,
Hmm.
transcriptional factors unique to the Y are conferring increased risk.
Hmm.
Does that mean that women or girls with female disorder have a different disease?
Fantastic question. So I think a corollary of everything I've said is, and if I go back to the beginning and say I'm imagining a future where in considering human biology we understand an XX biology and XY biology where they're the same and where they're different, I think that there will be cases in the future where we will discover that a disorder that has been given the same name in males and females are actually two different disorders. And so I think that's an really, and you know much better than I in the case of autism, there might even be some people thinking along those lines already. And certainly that, I can tell you that in the case of heart disease, there's increasing realization that male and female heart attacks are very different beasts. And so I think we need to open our eyes and our minds to these possibilities. Of course, therapeutically, there may be different, there may be great differences with regard to issues of safety and efficacy down the line. So all of those issues, I think, come into play. Yeah.
All right, well, let's give David a big hand.
Thank you.
Fantastic. That's fantastic.
That's a great talk.
Oh,
There
thanks.
must be mutations in those 12 genes in mice. Are they viable? If they
Oh.
are, do they have neurological phenotypes?
Well, so the crazy thing there is that
See, exactly. Yeah.
the evolution of the X and the Y in the mouse has gone further. So it turns out, actually, I'll just flip it back here for you.
So may not have those 12.
See if
I see.
yeah. So it turns out
There's a
mice
difference,
only have mice have a much more complete inactivation of the X chromosome.
I see. So
And they
the
have
mice are not
fewer
gonna be a good
surviving.
model for
So, and actually exo mice are fully viable. It's a problem. It's a
so mice
serious
is not
problem.
a model for the system at all. Then.
It's not. And that's why I say we really need to think about this in a broadly comparative way. We can't make any such assumptions. So
And there's no Y gene there's no gene that's evolved that's somehow unique to the Y chromosome.